Hi everyone, and welcome to another bioCADDIE external webinar. In today's presentation, we're going to have Carole Goebel. She's a professor in the School of Computer Science at the University of Manchester in UK. She leads a team of researchers and developers working in e-science, building infrastructure for researchers working at the lab. National and pan-national level. Her current research interests are in reproducible research, asset curation and preservation, semantic interoperability, knowledge exchange between scientists, and new models of scholarly communication. She's also a deputy director of the Elixir UK node and is a co-investigator of the UK Software Sustainability Institute. In today's talk, she's going to talk about the research object initiative, frameworks, and use cases. Please welcome Professor Carole Goebel.
Thank you very much, and thank you very much for inviting me to give this webinar. So I'm going to talk about research objects, and I'm really going to talk about the concepts. Then I'm going to talk about frameworks and use cases. First, I want to give it a sort of little bit of motivation. And the motivation, particularly from the work I'm doing on reproducibility, as you mentioned in the introduction, is based on the fact that we're moving away from manuscripts to the notion of research objects. And everybody on the bioCADDIE call will be probably familiar with that because the NIH program and bioCADDIE are very orientated around new models of being able to publish research, and in particular thinking about packaging together or bringing together the entire collection of activities needed in order to be able to reproduce research. So as David Donahue said, that an article about computational science is really to do with advertising the software and the data and the instructions associated with the research that the publication is actually a narrative about. And that means thinking about software algorithms, workflow scripts, all as first-class entities that we're producing as research products. The other thing that I'm going to be a common theme in my talk is that many of these assets are scattered, and I use the word asset here. I tend to use the word asset in our projects to mean a model, to mean a workflow, to mean a date, to mean a file. So the different kinds of research outcomes are the assets of that research. So we'll find code in GitHub and some materials, some data maybe in Figshare, and the slides that explain a particular finding will be in SlideShare and so on. And we'll also be using community databases. So the collection of objects that actually back a paper or an investigation may be spread in multiple repositories in different locations held by different communities, different hosts. So that's something I'm going to come back to. And one of the real pains we have in science is finding all of these different things. So we're probably all aware of this term FAIR, the findable, accessible, interoperable, and reusable meme for discovering these data assets and also bringing them together, as I've just described. So for example, there's Data Fairport, which is an initiative in the Netherlands. There's bioCADDIE itself as a data discovery index for the NIH. In the UK, we have the idea of a research data registry and discovery platform. There's this whole notion of being able to discover and bring together these particular assets. So let's go into a bit of the concept of research objects from my and my group's point of view. So really we had two main drivers. The first driver is our team have extensive and long experience of computational workflows and scripts. So these are sort of multi-step, nested, complex computational processes, which by their very nature are compound. So there are codes that we're executing, remote and local services that will be executed by these scripts, libraries associated with the executing platforms, the data that they generate, the data that they consume. We also know, for example, that these are, because they're compound, made up of many different parts, they decay. So preservation, workflow preservation was important to us because if one of or two of the three of these steps is a call to a third-party database, and that third-party database changes its interface or is no longer there, then the workflow fails. So that's of course impactful on reproducibility of being able to rerun this workflow. So that's the first thing that we wanted to worry about, was how do we describe these kind of complex objects so that they can be repaired and preserved. The second area of our work is in systems biology, where we have, in systems biology, where we're constructing, for example, metabolic pathways. We're building models of these pathways. We have construction data, validation data, predicted data, standard operating procedures for the laboratory, in the laboratory, to collect data in appropriate ways, the way we describe samples, all structured around investigations and studies and assays. So this is a structure, the ISA structure, that's particularly been promoted by Susanna Sansone at Oxford, who I know is part of the bioCADDIE community. And here we're interested in particular systems biology groups being able to exchange their investigations, where the investigations will be multi-component. So we'll have many models, many data around those models, standard operating procedures associated with those data as well, again for reproducibility. So those are the objects, but then there's community, the commons idea. So we have these workflows, but we also have a computational workflow commons that we've run for many years, called My Experiment, which is basically a place where projects and individuals can deposit and share workflows and find workflows and catalog them. So projects like the BioVeil program, which is a whole bunch of people or individuals. And we've also been developing, of course, along with the systems biology, a systems biology commons for modelers and experimentalists in systems biology, projects and programs around systems biology to provide a catalog of their research. And I'm going to show a few examples of that. This is called FAIRDom, and it's a kind of it's a pan-European initiative to manage the outcomes of research in systems biology done in Europe. So I'm going to show a few examples of that to make it a bit more concrete. So this is a screenshot of My Experiment, which is the workflow commons. And this is a paper that's been published by our Swedish colleague, Sonia. And this is a workflow pack. So there's files which have different data. There's a workflow here, which is a file that describes a particular executable script, which happens to be a workflow in the Taverna workflow language. There's an external link to an entry in a marine species public community database. There's another external link in here. So this is all packaging together all the different components to back this paper, the workflows, the data, and external links. This is that's what I mean by a commons. In our systems biology commons, which is called FAIRDom, which is a platform, it's built by on a platform called Seek, which I'll go into in a little bit details a bit later on. So this is an investigation into glucose metabolism in something. I think it's a parasite. I'm sure somebody on the call will know. And this is structured around the notion of an investigation, studies, and assays. So it's a bit small. You can't see it very well, so I've made it a bit bigger. So here this commons allows us to browse different projects and people and institutions and programs, and projects are organized into programs and so on. Experiments are organized around investigation, studies as assays, as I've said. And we have different assets, data files, models, standard operating procedures, presentations, and so on. And at the bottom here, we have, for this particular investigation, a person associated with a project, 2 studies associated with it, 14 data files, 13 models and 13 standard operating procedures and 21 assays were actually needed in this investigation. This investigation backs this paper. So this DOI resolves to the entire investigation. So here we're thinking about not just something that's citable, which is a data file, or citable that is a piece of code, but the entire package, the entire investigation is citable, and this platform, the FAIRDOM platform, is effectively acting as a companion site for the published paper pointing to the investigation. So it's a commons in that regard. Here's an advert. So if you're interested in reproducible and citable data, we'll be running a workshop on this in September in Germany, and we'll come along, and it's mainly targeted at systems biologists, but we can make your data citable. Anyway, that's my end of my advert. So the SEEK system is the system that we use to build the FAIRDOM platform that I just showed, that commons. And I put this picture here because this seemed to me when I was preparing this talk and I was looking at the NIH Commons, that this is very much what we've already built in the systems biology community. So we have we support different research infrastructures. For example, people can use Dropbox, things like this, in order to be able to deposit things. We support tools, so you're able to simulate models that are uploaded to the Commons. It has very strong links to public repositories, so you can cross-link to standard model repositories or data repositories, and so on. So it seems like this was very synergistic to what's going on in the NIH and bioCADDIE. And so the idea here is that you're able to catalog and search and index outcomes from the different systems biology projects advising in European programs. All right, so that gives you some idea of my sort of background. And from this, we can kind of emerge 4 major concepts for our research objects. The first is they're multivarious, citable research products, as I've just shown. So that DOI is a citable research product, which is a complex product. So this recognizes the fact that code and models and standard operating procedures and data entries should all be valued and cited. The second, and this is very important, is they're compound, and they're nested, and they're scattered, but they're interconnected. So this is a bit more of a schematic view of the systems biology SEEK platform that I just previously showed. So this really shows the investigation, studies, and assay structure. And so these components link out to local repositories or link out to its own private own centralized store, but also to PubMed, to the external databases, to model platforms. So this is basically a spanning Uber index, bridging across these various different repositories where data and models and code and all these different assets will be held. So not everything is in one place, it's scattered. And that's very important later on. The third thing is that we're interested in portability of research products into platform exchange and reproducibility. So we've got many different platforms, so could have different SEEK or Figshare. So we may have something in our systems biology platform that then we need to be able to package up and put into, say, Figshare to back a publication, because that's what a publisher wants to be able to use. So this idea of being able to ship things around different platforms as well as different laboratories is an important one. So we can either have different platforms and different communities, or we can try to create a commons so everybody puts their stuff together in one place. So there's a mixture there. And in particular for these kind of compound artifacts-like workflows, we want to be able to check things like a checklist of Are all the definitions there? Are all the services still accessible? Is the input data present there? So we have some notion of being able to describe these sufficiently so that we're able to rebuild and repair these workflows when they break. So this notion of packaging things together and being able to port things is very important from a reproducibility point of view. And then the fourth thing is that the notion of active versus snapshots. So these research products, as research evolves, so do the products evolve. So this is a schematic from Martin Sharm, who's on the call, I think. Hello, Martin. He's a systems biologist from Rostock, and he's particularly been investigating the evolution of models. So these are mathematical models that describe metabolic pathways, and so they fork and merge over time as science forks and merges over time. So it's important to think about versioning. What does it mean to cite something that's constantly being evolved? So there's this difference between a research object that's kind of living, that you're referring to, and the snapshotting of research objects so that you can fix it so that it can be citable to defend what you knew at a particular time, perhaps to back a publication. So these are four main concepts that kind of emerge from our ideas. So when I was thinking about this, I was thinking about, well, what's the metaphor for something that is spans many different resources. It's constantly, it may not, it's kind of a box where you're linking out to things that are evolving in time, things that may not be there because they're actually remote. You're just indicators to them, so they're not all within the box. They're kind of outside of the box. And of course, for those geeks of you on the call, you'll be familiar with the notion of a TARDIS, which is a time and relative dimension in space time ship, which is used by Doctor Who to travel between time and space. And this is what I kind of thinking about for research objects, that the content ranges from closed and embedded and fixed and local to open and fluid and remote. And they have multiple authors, multiple stewards, multiple sites, and they kind of span over time, and they span over platforms, and they span over researchers. For those of you who aren't TARDIS geeks, all you need to know is that a TARDIS is bigger on the inside than on the outside. That's the important thing about a TARDIS. When you go inside it, it expands, and this is what a research object is the same as. When you go inside it, it expands because it links out to things that are outside it. I'm sort of waving my hands about, which is no good on a webinar, but never mind. You can imagine me waving my hands about. So this brings together my framework for doing what we might call knowledge turning in science, which is moving knowledge through the process of a scientific publication and sharing and processing. And so I would say that we're interested in transferring things between people through a commons. We're interested in releasing things over time, and we're interested in interpreting things in order to make them reproducible. And for that, we need research objects. And I'll go into the more details about research objects, what we mean by research objects, framework there. And for those of you who are astute, you'll see that normally when we develop these frameworks, we have pyramids and we have layers. Well, I've done a TARDIS. So this is a framework that is based on science fiction. Okay, so let's go into the research object framework in a bit more detail. So really it comes down to two major activities. One, how are we going to describe various different products? And we're going to recognize that they're running on various different platforms. And two, and this is the most important bit that I'm really going to focus on in this talk, is how do we bundle and port and link together resources? So how do we package together these objects so that they carry the research context of their combination, and create these as units of exchange rather than the individual data files or the individual code or other resource. So we needed a few desiderata. So the metadata has to be machine processable if at all possible, but also we have to have a way of people being able to read it. We should use standards. We should try to develop something that was technology independent. It should work with multiple platforms. And most importantly, we should do the least that it's absolutely possible to do and get away with using standard tooling and allow incremental buy-in so that things would gracefully degrade. So we couldn't just do all or nothing, that we would try to do an approach that was just enough, just in time, as opposed to just in case. So in order to be able to take on easy or at least straightforward if it wasn't easy. So to do this, we came up with a core model which uses some standards, which I'll go through now, which we can then use in order to be able to describe what we call annotation profiles, which are descriptions of the metadata of a research object. And then we have metadata formats that we can express this core model in, because what we're talking about with the research object is a research object really is about the metadata that describes each of the objects and describes the package of the objects, the grouping of these objects, and an API specification. And then we need to actually map this to some really practical systems. So we really have to implement it. So we're following strongly the sort of standards route, which says it's okay to have a specification, but you must have an implementation. You must show how this will work with commodity systems, or how you may migrate something, a platform, a commons platform like the SEEK platform, in order to be able to make it research object compliant or aware. And then there's a whole bunch of things around policies and service and stewardship and training, which I'm not going to go into this talk. So I'm just going to run through this now. So the core model is based, again, we're trying to be very, very simple here, on three basic premises: identity, so you need to be able to refer to things; a metadata description that has really two big roles. One, what is the objects, descriptions about the objects, and how they are linked together; and two, attribution. So this is provenance. This is the who and when and why and where of the objects that we're trying to describe. And then the third thing you need is aggregation, the ability to group things together, to group the constituents together, and to talk about the group of things. And that means when you're grouping, you may be grouping things that are physically there, or you may be grouping things that are virtually there, because you only have their identifier, but you can talk about them, because that identifier resolves to, say, an entry in GitHub or an entry into a public database. So to do this, we went into the standards route. So of course, identity, there are numerous kinds of things that one can use, most of them based on the handles approach or URIs. We used the notion of annotation, so a way of being able to just say, I am describing a series of, I'm building up a series of annotations, so I'm just doing sort of standoff annotation, associating a description with an object. And this gives you a point of extensibility. There's a standard for that, or an emerging standard called the Open Annotation Data Model. So this is a way of being able to associate annotations with an entity in a standard kind of way. And then aggregation, which is a way of being able to, the metadata for describing how you group things. So this is all metadata. This is all to do with how do I describe research objects. And I'll show you how we're going to be using that in a minute. And these are standards. So the ORE is a standard from the library community as a way of describing groups of things. And there are platforms then that we're going to later on map and use in reality for these particular metadata descriptions. So I'll be going on to those in a minute. So there's a difference between the way that we describe the metadata and then how we actually deploy that into the platforms, the computing platforms that are available to us. So this is just, I don't expect you to read it, or if you can read it and debug it, you're a better person than I am. This is a research object model ontology that is basically the smallest amount that we could get away with for being able to organize the concepts of aggregation and annotation. And it's used in something called a manifest. So that's the bit I'm going to do next. So what we have with our research objects is the objects that we're going to be putting together. This is all of our different bits and pieces of our experiments. And we're putting them together in a notional package which we describe with a manifest. So these objects, they will themselves have descriptions associated with them, which we can use the research object model to associate those descriptions with them. I'll go into that a little bit more in a minute. But importantly, the whole packaging together of these, grouping them together, is described by this manifest, that describes the content and the relationship of the content. And here we're really talking about the title and the creator and the status of the object, the whole thing, a way of being able to aggregate the whole thing, and the annotations that describe the things. So this is what this manifest is allowing us to do. So it's packaging everything together. So the manifest is the critical piece. So here's a very, very simplistic manifest. So this is my research object. It has this ID. Here's some metadata about it. And then we're going to have with it some aggregates. This is the things that we're going to be grouping together. And then annotations, a series of different files that describe or ways of describing, say, in this particular case, it's a workflow manifest. So this describes the results, the provenance of the workflow using, in this particular case, the RDF format. Specific metadata associated with it that is domain specific, i.e. that is, say, biology or systems biology, which is in a JSON format, and something that's actually in a readable format for a person. So we're kind of associating these annotations are associated with the objects. So that's the metadata, how we're going to be doing that. Then we have to actually deploy this using off-the-shelf tools for being able to organize containers. And there are really two classes of tools. There's things that we might call catalogs and commons. So the FAIRDom Seek system that I just presented earlier is a commons. It's a platform that effectively is inherently grouping things together. And remember, these groups can be nested, of course. But there are also other platforms for containers, which are for packaging things, notably zip files and things like Docker images. And these are techniques for being able to package things together so that they can be exported. And so they really are container platforms. And we use both of these for the research objects. So we've got a specification of the research object bundle. And this is a way of being able to bundle together, export, archive, and publish and transfer our research objects. So this research object bundle is a file format for storage and distribution of these research objects as zip archives. So zip is the least you could do. It's the simplest thing to do. So you can include in the zip folder, zip archive, the research objects manifest, all the annotations, and the aggregated resources, some of which will be physically there and some of which will be pointing to other resources. And it's the basics for other formats. This is the basis for other formats that you can extend. And of course, it's backwards compatible because it's a zip file, and we have programmatic access as well, because we can represent the, as I said, shown before, the manifest as JSON or JSON LD, for example, linked data, and we have APIs to this. So this is this is what it would physically look like. So here's the research object. It's the zip file. It's got a manifest. It's got a descriptions. Here's the file folder with all of the descriptions of the annotations, and here's the actual data. So we're packaging everything together. This actually raises some interesting issues as well, by the way, which is if we have these evolving research objects, which are packages of things, then we also have to worry about identity and resolution. And this is an interesting topic for the interested reader, because as I've suggested, that the key thing here is identification, that we have to differentiate with our research objects as they kind of evolve from, are we using a research object to defend our research, or to locate research object, or to reuse it, or to credit it, or to cross-link with it. And so the identifier, and it's really the identifier which is at the heart of this. If you can't get the notion of an ID sorted, you're pretty screwed. The identifier and its behaviors is pretty important. So how do we allocate identifiers? How do we fix identifiers? And how we resolve these is an interesting question, which we have various different techniques of being able to deal with. But it's really this notion of this very fluid research object, this TARDIS-like research object, does raise issues to do with identification and life cycles, which I won't go into at the moment. So, okay, so now we've got this notion, we've got a way of describing things, a model for being able to describe things and be able to group things together from a metadata point of view. So what is the metadata that we're going to be describing? So, what are we going to be putting in our annotations? And we really think there are four major things that are absolutely critical: provenance, versioning information, dependencies, and checklists. So provenance is fairly straightforward, and W3C Prov is the system that we typically, the standard we typically use for being able to represent provenance. Versioning, there's GitHub is well known for its versioning. There's all sorts of dependency models for being able to implement the dependencies. So how do I represent dependencies like Puppet and Makefile? But Docker is one of the ones that we're particularly, a platform that I'll mention a little bit later on, that is a powerful platform in this regard. And but the interesting one is the checklists, which is something that's kind of well known from the life science community. So the life science community has for a long time developed these minimum information models, which are effectively checklists of what you collect in order to be able to report on an experiment. So this is the minimum information about an RNA AI experiment. But we have all of these for microarray data, for proteomic data, for all sorts of checklists for basically what am I collecting when I'm describing a particular kind of data. And we also have these checklists for systems biology models as well. What do I need to be able to record? What do the parameters do? So on. We also have them for workflows. What do I need to be able to record of a workflow? Do I need to be able to record its input data, and so on? So checklists are a prime way of being able to say this is the annotations that should be associated, one, with the actual object itself in the repository, and in the research object, and two, in the research object package in the manifest. So the checklist describes, say, a microarray data file, and the checklist can also describe what needs to be in the research object in order for it to be valid. Like that microarray data may have to, for example, link to, I don't know, standard operating procedure that it's associated with. So, and we expect these annotation profiles to become very richer as you go out of this kind of cycle of annotations, and to become much thinner as you enter in. So one can imagine, for example, one can imagine it's true, that at the center, the minimum you would have is citation information. And then you'll go out to doubling core for the library kind of, who's done it and at what time, out to experiments, and then out to type specific. So for example, descriptions associated with workflows or descriptions associated with systems biology. So it kind of becomes richer as you expand on these annotation profiles. And the annotation model of the research object allows you to extend these types to be more rich. Okay, so let's, in the last 10 minutes, do the use cases. Okay, so the first use case is our Systems Biology Commons, as I've already mentioned earlier on. This actually is a native research object platform, so at its very heart it has this notion of highly structured and interconnected components which you can then package and label and extend. But it can also export and import research object bundles, the kind that I've mentioned as zip files. Its structure, as I think I've already mentioned, is based around this ISA platform, Investigation, Study and Assay, which Susanna Sansone's been working on. And we have a just enough results model, which describes just enough of the results of systems biology experiments with the common elements and relationships to be able to register them and exchange them. And this is the metadata that we use for, this is the structure of our packaged research objects in the SEEK system, in the FAIRDom system. And here we have the notion of living research objects. So this is a living object that is in the platform. But if we want to snapshot it in order to give it a DOI and to point to it from a paper, then we can snapshot it and retain it in this platform, or we can snapshot it and export it as a package, as a zip file to deposit into, say, Figshare. There's also some standards associated with systems biology. Notably, there's something called Combine, which is an archive standard, which is a file that contains all the information to describe a systems biology model and simulation experiment, i.e. the pile of maths that you need, and the ordinary differential equations and the simulation and the data associated with it, and the parameters, how the parameters are set, all marked up in a standard markup language called Systems Biology Markup Language, surprisingly. And we have mapped this and combined this archive standard with the research object model, which has the benefit of standardizing how the meta is described, standardizing the API, and regularizing the provenance that's associated with that legacy standard. And then what we're able to do then is to say, okay, so we have a commons of systems biology. This is our commons platform. Then we can export as a research object. We can incorporate the Combine file as part of that, because it's also a research object, using all of the different standards, and pop it into, say, Figshare or GitHub in order to be able to publish it. Also pull it out again and put it into another Seek installed somewhere else for another community in systems biology, and they can unpack it and have a look at it. So we can use it as an exchange mechanism. So this is where we're getting the exchange idea as well as the commons idea. And so this kind of puts it more graphically. So we export it as a zip file, we put it into Figshare. Here you can unpack it. You can't see it very well, of course, because it's very small. And then here's the actual files. And in fact, I've expanded that a bit so you can see it really. I'm not making this up. It's all true. So here's the manifest, and here are all the data files and the investigations and the descriptions and so on, and all the models and so on. So the whole thing's packed together, and then you can unpack it and index it and catalog it. And here's what the RDF looks like in the manifest that describes how everything is held together using this just enough results model. And so you can see this is all describing the different pieces and how they all link together. And you can query it because it's described in this particular case in RDF. So you can do a query over it to find all the different components. So it's a very rich metadata object. The second case study is our Taverna. I won't go into much detail on the other case studies. Is Taverna workflows, these complex scripting environments. And here we can use the research object approach to bundle up the run of a workflow. So we can use it to be able to describe the workflow, the inputs, the outputs, the provenance run, the intermediate files, and all the different and links to who ran it and which resources were used in order to be able to execute that workflow, particularly if they're remote resources or third-party hosted resources. So we can again use the same idea in order to be able to describe and package up all of the different parts of that workflow when it was actually run. And so the potential here is that we can use the research object idea in order to be able to kind of have a stepped view. We can use it to package up the actual workflow system itself. We can use it to describe the workflow example configurations, the different plugins and versions of the workflow system, of the workflow description. The actual workflow runs themselves. So we kind of can build up different layers of descriptions of these research objects, and then we can use the packaging to deposit them or to exchange them into various different community type of repositories. The third use case we're working on at the moment is with the Atlas Collider data analytics. This is a work with Charles Vardaman and Daho in Notre Dame, and they're interested in dealing with all the files and data for executing experiments on the Atlas detector, the Large Hadron Collider experiments. So they actually have a way of really being able to kind of build almost a virtual machine of what's actually happening on when the execution of the analytics is being done. They want to be able to package that all together, and they use Docker to be able to do that. Docker is this portable, lightweight application runtime packaging tool, which is the Lord's work. It's great stuff. But the images that it produces have metadata descriptions, but they are a bit opaque, shall we say. So if we use this as a packaging mechanism and then use the research object bundle to describe better what's in the Docker image, then it's much easier to understand what are all of these different components that have been brought together, and therefore it's easier to read and it's easier to run, and we can also package it all nicely and put it into a third-party repository like Zenodo. So we can relate all the different files and the layers and put provenance and annotations and all the things that Docker doesn't do. Docker is really pulling together just the dependency layers of the different pieces of software and the instructions, the different pieces of software largely in the data of this experiment. And then our final use case is the FAR Institute Commons. The FAR Institute Commons is, or the FAR Institute is a multi-site institute funded by the Medical Research Council in the United Kingdom, and it's basically interested in the safe use of patient and research data for medical research. So it's basically interested in how to use patient data in the wild for being able to assemble clinical study cohorts and be able to do clinical research. So we have the glorious National Health Service, which has an enormous potential for being able to establish those patient records for research. And this is what this is about. And it's particularly, and here the research objects are data, samples, scripts, analytics, longitudinal studies, different laboratories with different software platforms in different parts of the country, and in Scotland as well, which is almost a different country in the UK, and lots of legacy data. So how do we exchange resources between them? And we do it by research objects, of course. So each of these laboratories are running their own ways of being able to organize their data, but they can map and package their data together, their data and their scripts and their publications and so on, and then ship it between one another using this standard mechanism. We're also building a commons based on CCAN in order to be able to catalog and index these as well, but not always. Sometimes you just have to privately share things because of the nature of the data, and you can't always ship it either. So this is a kind of screenshot of the commons that we're building. So to conclude, we're building, we need some infrastructure, of course. So to make and import and export and inspect these research objects. So we've built some libraries. Again, we're trying to do the very least, which is to create infrastructure so other people can include this kind of approach into their own platforms. So we're not trying to build a large-scale infrastructure here. So we've got Java, Ruby, and Python libraries for creating and inspecting the research object bundles and their metadata manifests. We have built some user tools. So we have a command line tool for making research objects. Command line tools are great. And we did a research object hub prototype web application to manage them, but command line tools are not to be sniffed at. And we also we're also building two platforms, or we have two platforms that are research object orientated, if you will, the SEEK, as I've already explained quite a bit, and CCAN, which we're finishing off a plugin to build and import and export research objects into CCAN. So to conclude, to wrap up, I was looking at the NIH Big Data to Knowledge kind of commons conceptogram, as it were, and this is the one that I found on that's on the web. And I was looking at how does our research object ideas map to that? Well, we have these metadata profiles, which I try to explain for discoverability and findability. We have the research object model APIs. We have we've done a lot of work on community IDs, which I haven't really explained, but if you're interested in community IDs, I recommend looking at the Biomed Bridges 10 Rules for Identifiers, which is really good. The container piece, the piece about containing, packaging, we really spent a lot of time on that, this research object model manifest profile, and we spent a lot of time really thinking about how do you implement it using off-the-shelf techniques and off-the-shelf technologies. So in summary, what I hope to have got across to you is that we've been working on these findable, accessible, interoperable, reusable research objects. I've presented the concept, the model, framework, and the use cases, some use cases, and I hope you've got the idea that it's kind of very lightweight and incremental. The idea is to be simple and extensible. There are significant challenges with the whole vision. The two big ones, I think, are multi-stewarding and life cycle. What does it mean to life cycle these research objects, and what does it mean when you have multi-stewarded, i.e. changing resources that you're linking to in your research object, because that inherently is changing the research object, because you're not entirely stewarding unless you fix it and snapshot it, and that's why we've been looking at fixing and snapshotting and packaging research objects. And we're currently building partnerships with Figshare, with Oxford Bodleian Laboratory, with the Far Institute, and it'd be interesting to see if we could do something with bioCADDIE. Okay, so that's it, and these are my acknowledgements for all these people who've been working on research objects and people and various different sources of funding. Is anybody still there?
So we're going to unmute all the lines right now and then open for
The
questions.
conference has been unmuted.
Okay, so questions for Professor Gomo?
Hi, this is Heidi Sophia. That was a fantastic talk. Can you tell us a little more about, how you might intersect with bioCADDIE?
Well, I'm here to find out. I'm excited and looking forward to next week's metadata working group, which I think is having a call. And we've also been invited into the identifiers working group, which I don't think has had a call yet. And I'm also talking to Susanna Sansone, who of course I work with a great deal, and she's been, as you can tell, a big influence on the ISA structure, which has been very powerful for us. I guess I'm kind of looking at all the bioCADDIE white paper and literature I've been able to identify. It seems that you've really this commons idea. We spent a lot of time. We've actually built commonses, and they're actually being commonses. Is that a word? I don't know. Anyway, I've just invented a plural called commonses, and we've had a lot of experience doing that, and we are still running those platforms. In fact, we're heavily funded to continue to do it. So we may be able to bring our experiences to bear. I think the other thing is the model for describing metadata, trying to keep things really simple and extensible, and to bring that to bioCADDIE, I think would be useful. But I'm still feeling my way a bit. Do you have ideas?
Well, that all sounds very interesting, and it'll be very interesting to kind of see how this come to the table. There's one little cultural element that I wish that you would bring, and that is the practice of the stickers. You know what I'm talking about? The stickers referring to your software.
The stickers. Oh, yeah, the stickers.
Yeah.
I have stickers. Yeah. Well, if I was physically there, I could see you all.
Could you explain the meme of the stickers? Because
I would be forcing you to have stickers. So, all my software have stickers associated with them, and I stick them on everybody, including everybody's laptop and so on. We also do T-shirts, and stickers are an incredibly powerful meme. You can get so your ambition is that your funding agencies will all have stickers on their laptop advertising your software. I found this to be is viral marketing. Viral marketing.
I have to attest to the success of this because about 4 years ago you gave me one of these stickers, and I just thought it was a brilliant idea, and I think it works exactly the way you describe. It's a viral marketing and it has a meme-like effect.
Yes. So I'm all for viral marketing. We're in the business of actually sales. We might think we're academics. Actually, we're salespeople because we're selling ideas and concepts. And we have to use exactly the same techniques as salesmen. Actually, if I was giving this talk physically to you all, I'd be wearing my TARDIS dress. I do own a TARDIS dress. That shows you how geek I am.
Well, it's good to see you maintain a high standard.
Thank you. Are there any other questions?
Thanks, Carol. This is Ian Four. Just to pick up on the bioCADDIE connection and that metadata working group that you referred to, I think the levels of progression, concentric diagram concept, I think is a useful one to guide that metadata discussion, because I think it has the idea of core metadata and then pushing outwards to more detailed metadata, which can answer some very sophisticated questions. But I think one of the balancing acts we have to do is to work out how far in each direction, and I think you had 4 directions on your diagram, basically, though, you know, there may be some which are sort of a little bit mixed, but I think it's a useful way to, at least a useful framework to say, be asking the question, well, how far are we pushing ourselves out there towards the perimeter, and what is the cost of doing that? And also, how can we get further out? Because a lot of the real value lies the further out you can get there. But I think a lot of the time you can't get out there on your own. You've got to do this by using a framework that everybody else is using, and different people can fill in parts of that diagram. Nobody can do the whole thing.
That's absolutely critical, Ian, because the important thing here is to do with being gracefully able to degrade as well. So the idea is that if the only thing you know is some basic doubling core of something, you can still do something with it. Or if the only thing that you can understand is doubling core, then you can still do something with it. But if you know more, or there is more, you can do more. The idea was to make it so that it was really incremental, and so the more capability that you had, the more you'll be able to use. But you wouldn't be forced to do everything, because this is death if you do everything. If you make a model that is so comprehensive but also mandatory. We've seen this in other systems, particularly ambitious kind of cataloging systems that try to make everything be recorded. You just don't get anything. So instead you have to really go for this kind of incremental approach. And that means building a framework to be able to do that incrementality. And that's what the core model of the research object allows you to do. It's a way of being able to express that the metadata in an incremental way, but in a standard way that the tooling can actually understand and unpack. And that, I think that's absolutely the key thing. All using standards, all using commodity platforms.
Thanks.
So hi, Carol. This is Susanna.
Hello, Susanna. I just did a big push for you.
No, thank you. I've listened to it, and actually Ian was faster than me to follow up to ID comments. So yes, also for the benefit of the other, Carol is in the metadata working group, which indeed has the first call next week. And we really look forward, indeed, Carol, to discuss how we can use the core RO elements. So we will have some work which we will show to you, but we need your input in this particular, as you were telling here. How do we actually have a core and we build the extension in a progressive way, but in a very sensible and pragmatic way too. So thank you very much and look forward to it.
Oh, thank you. And Stian Solanderis will be joining me, and he's one of the key people who's built the infrastructure. So I think we'll be good. I also think another big thing that I think Heidi or Ian asked, what do we also bring? Implementation is really important, to not just come up with ideas, but to also come up with commensurate implementations. Because if you don't have that, then you can't really demonstrate what you're doing. We took this from the W3C standardization activity that says, if you do a spec, and you'll see our research object bundle spec looks stunningly like a W3C spec, because that's what it's modeled on, and I would really strongly recommend following those kinds of principles and that kind of template. Then you need also implementations to go with it, and I think it's a very good idea. So that kind of rigor is quite important too.
Absolutely, and bioCADDIE has a developer team which somehow implements what the different working group will do. Although next week we indeed are going to discuss the spec rather than implementation, but hopefully some of the development team can also follow our code. There are so many codes in parallel, but
there are. It's hard to keep track. Yes.
Yeah, and align all this with Elixir too, of course.
Yeah, that's an extra complication.
Indeed.
Yes.
Hi, Carol. This is Jeffrey Greta.
Hello.
Hello. And one question, for how, getting back to how this could potentially relate to bioCADDIE as well as, how are you currently tracking sort of, the different instances of the research objects that might be in different places that have, perhaps some further analysis was done in one place, or was it shipped somewhere else where it's also being made available? So is there, is there a use
You mean
case
like a notification kind of thing? Oh, something's changed here. That kind of
where
thing,
does because as these research objects, and data sets in general get placed in multiple places and used by multiple people, you sort of have then multiple instances of the same entity, existing in different places, and being slightly changed. So how do you, how do you keep track of those differences or changes, or is that something you're doing? Because I could potentially see that as something that a bioCADDIE DDI would be able to help with.
Yeah. So I see what you mean. So this kind of comes down to. Let me take the example of our systems biology commons, what's called the FAIRDOM Hub, which is built using the Seq computing platform. That is a single place. It's that you can record and deposit, basically support your research objects as you're doing your research. And there, if things get changed and evolved, it's a central place. You can see it. It's all in one place.
Right.
As soon as you start to package things up and ship them, unless you've kind of got a tether to them, then they're kind of out on their own. And this is what's kind of interesting, because then it becomes an issue to do with, well, am I fixing them and completely materializing them because I want them to be fixed? Because they're, for example, going to be deposited somewhere in order to be able to defend a paper when you don't want it changing, or is it going to be a link back, or are they not going to be fully materialized? Are they going to actually be almost semi-virtual, so that what they're based on is evolving, and therefore inherently they will be evolving as well? So it's kind of automatic that they are changing because they are effectively semi-virtual objects. And then there's a kind of probably a third version, which is what you're thinking of, I think, Jeff, which is where you've kind of materialized them, but their originating sort of resource is changing, and now they're out of sync. Is that what you're thinking?
Yeah, I think it's both 2 and 3, right? So that you have a research object that you published, and now I'm extending the model by adding a new channel type or something of the nature.
Yeah.
So I still have the original research object, but now it's, the provenance says I come from research object 1, but I'm actually research object 2. So you have these research objects now in different contexts, scattered.
Yeah, so it's a versioning. There's a versioning idea of the research objects, and the provenance of the research objects, and we know how to do that. But I think what you're proposing is something slightly more sophisticated or different, which is how do you know? How do you keep them in touch with one another? So that, because you can say that I'm evolving one, but that doesn't necessarily mean that you're then reporting that you evolved it to its originator.
Yes.
And
exactly.
That's a notification activity, and I think that's we've done some experiments in that, but we haven't actually built any kind of real, what I would call, production type of stuff.
Yeah. Because that's really an identification, identifying the previous research objects, and then because you'd really like to know, how people have extended certain models or Or
Indeed. And when's your identifiers working group meeting, Jeff?
Well, we had the first one this morning.
And
then the next one in 2 weeks. So.
Right. Okay. So we'll make sure that we're on the call, because this morning I wasn't around. Right. In 2 weeks.
Yeah, I'll make sure, Carol, this is Stephanie, that you get the invite.
Thank you. Yeah, I don't think I got the ping about that.
No, I think I added John and didn't know you were going to be on it. You wanted to be on it also.
I think Stian's signed up to it. And,
yeah.
Carol,
I'll make sure you get back on.
Thanks.
And just for information, Carol, Julie was on the call as well, so she voted in the work. Yes, don't worry.
Oh, good, because Julie has forgotten more about identifiers than most people know. That's good. But I think that's a very, very interesting question. And if you recollect, Jeff, one of the things I raised during the talk and also at the end was this notion about life cycles and what does it mean with the identifiers. And that, I think, is the biggest challenge. So that would be good to work with bioCADDIE on that issue.
Okay. I think we end up a little bit late in the 1 hour presentation that we have here, but I think we have a great group of discussions. I would like to thank Professor Gumbo for this great presentation today. And then I also have an announcement for the next external webinar. We have a change of the date. So it's going to take place on July 2nd instead of July 9th. And Dr. Hwanjo Kim is going to talk about new challenges in big data, technical perspectives.
Ooh.
So thank you, everyone.
Okay. Thank you. Thank you, everybody.
Thank you.
Thank you.
Bye.
Bye.
Bye.